Okay. Good evening. Welcome to our internal exam marathon series. So today we'll be beginning with our vitamins. So I've divided the vitamins into two, fat soluble and water soluble for the sake of convenience. So we'll be doing fat soluble vitamins today. That is A, D, E, K. So I'll try to cover all of them. If any of it is left, we'll continue with them in the next class and then move forward. Okay. So introducing you to the core features of Oracle. So here at Oracle, we have live classes to elevate your learning experience. Then you have recorded classes as well, where you can assess the recorded lectures as per your convenience, anytime, anywhere. And apart from that, we also provide you with notes and MCQs based on the latest syllabus to assess your knowledge. Then a very unique feature of Oracle is that we also give personalized counseling sessions. So anytime if you feel that you need some kind of extra support and guidance from our end, feel free to book a slot with us. Then we have three different subscription plans at Oracle. That is the basic, which is for 3 months and it comes for 15,000. Then the standard plan, which is for 6 months and comes at 30,000. And the premium plan, which is for 12 months and will cost you 50,000. Apart from that, we have 20% discount also if you use my coupon code as displayed here on the screen. Then briefly telling you about the internal exam marathon. So this series has been specifically curated to target your university exams. So we'll be doing rapid revision of all important questions and frequently asked topics in this. Volatile quick facts will also be covered. Then you have multiple choice questions so that we can assure you top-notch grades. So before I begin with the individual vitamins, I'm giving a very brief introduction regarding vitamins so that you have an idea what vitamins are and about their classification. Okay, before we begin. So introducing you to vitamins, these are organic compounds which are present in small amounts in various food substances needed for growth and maintenance of the body. So first thing that these are organic compounds and they are present in very small, minute amounts in various food substances. Okay, so basically they have to be supplemented through diet. Okay, later in the lecture, I'll tell you which of them are endogenously synthesized. But in general, they are usually supplemented through diet and they are needed for the proper growth and maintenance of the body. Now, vitamins are dietary essential, okay? The body does not synthesize vitamins with the exception that certain vitamins though are endogenously synthesized by the body. So usually they are supplemented through diet, but some of them, like exceptions, biology is a science of exceptions, we all know. So some of them, they are endogenously synthesized by the body. Now, endogenously synthesized vitamins, there are two types, that is synthesized by the body and synthesized in the body. Okay, so our body is synthesizing these vitamins. Okay, so the one that are synthesized by the body, they are being synthesized by our body itself. But in the body means that somewhere inside the body, someone else is synthesizing those body. So the synthesis, although it's taking place in your body, but it is not taking place by your body. So that is the difference between the two. It is by the body and in the body. Now, synthesized by the body, the example is niacin, which is derived from tryptophan. If you remember, when we were doing tryptophan metabolism, we had discussed the NAD pathway as well. So niacin, that is synthesized from tryptophan. A vitamin D, that is synthesized from 7-dehydrocholesterol by exposure to UVB rays in the 920 to 315 nanometer in the skin. Now, in the body is your vitamin K, biotin, and pantothenic acid. They are synthesized by the bacterial flora in the intestine. So, these vitamins, that is your vitamin K, biotin, and pantothenic acid, they are synthesized in the body, but by the bacterial flora present in the intestine. So if there is a question which mentions like which of the following vitamins are endogenously synthesized vitamins, and you have in the example niacin, and you also have biotin. So preference will be given to the one that is synthesized by the body. So your answer will be niacin and not biotin. So moving on to the classification. So they classified into fat soluble and water soluble. This is the most easy classification. In your fat soluble, you have vitamin A, D, E, and K. And in water soluble vitamin, you have the entire B complex plus vitamin C. Now, a little comparison between the fat soluble and water soluble vitamins. So, your fat soluble vitamins, they are absorbed as chylomicrons because they are fat soluble, okay? So, they cannot be directly absorbed. Whereas, your water soluble vitamins, they do not need chylomicrons because it can be directly absorbed from the intestine. Can be directly absorbed. Okay. Whereas your fat soluble vitamins, we all know that chylomicrons, they were responsible for carrying dietary tags. Okay. So since your fat soluble vitamins, they're also exogenous source, mainly from you getting them from exogenously. So they will also be carried through chylomicrons. Now the storage, fat soluble vitamins are stored in liver and adipose tissue, whereas your water soluble vitamins, they are not stored. So if something which is stored, it will not be excreted. So your fat soluble vitamins are not excreted, whereas water soluble, they are excreted in urine. Now, fat soluble vitamins, they do have toxicity. They are toxic since they are stored in the body and not excreted. Whereas your water soluble vitamins, since they are excreted, they are not toxic, with the only exception of vitamin B6 and niacin. And function, your fat soluble vitamin, they have varied functions. We'll study about each of them when we take them individually. And your water soluble vitamins, they usually play the role of coenzyme, so they have coenzyme function. In your fat soluble vitamin, the only exception is vitamin K, which is a fat soluble vitamin but has a coenzyme role. Okay. Now, talking about vitamin A. So, let's begin with vitamin A, then we'll take up vitamin D, then E, and K in that order. So, talking about vitamin A, it includes both your retinoids and your carotenoids. They are included in vitamin A. So, talking about retinoids, the active, they are the active form of vitamin A, that is your retinoids, and they are obtained from animal sources. Compounds which are chemically related to retinol, they are included in your retinoids, and your retinoids can have different forms, that is retinol, retinol, and retinoic acid. So your retinol, it has alcohol as the functional group, retinol is the aldehydic form, and retinoic acid, it has carboxylic acid group. Now talking about carotenoids, they are provitamins. Okay, so that is, it is not the active form of vitamin A, it is the provitamin A. It can be converted to the active form. Can be converted. Okay. And your carotenoids, they are derived from plant sources. Most prevalent carotenoid is your beta carotene. Its richest source of beta carotene is carrot. Okay. Now, there are about 600 carotenoids, the most important one being beta carotene, which is an antioxidant. Although, in fact, all carotenoids, they have antioxidant role to play, but specifically talking about beta carotene, so it is an antioxidant. Your lutein and zeaxanthin, they are both used in the treatment of macular degeneration. And your lycopene, it is used in the treatment of prostate cancer. Okay, so that was about retinoids and carotenoids. Now talking about the structure of vitamin A. So your vitamin A, it has beta ion ring, single, that is one beta ion ring, and an isoprenoid chain. What is an isoprenoid chain? It is a 5-carbon unit, okay, isoprene unit. Looks something like this, okay, as you can see here. So this is your isoprenoid chain, and this here is your beta ion ring. So this forms the structure of vitamin A. But your beta carotene, it has two beta ion ring. You can see here 1 and 2. Okay, and this is the site of cleavage. We will see when dioxygenase enzyme acts on this beta carotene. This is the site of cleavage, and it will split it into retinol. Now, the three forms, they can be interconverted. The retinol, via the action of reductase, can either be converted to retinol, and the retinol can also be converted back into retinol. Then your retinol, the aldehyde form, it can also be changed into the acidic form, that is retinoic acid. Now talking about the metabolism of vitamin A. So we'll take the metabolism of vitamin A under three headings. That is its absorption. How is it absorbed from the intestinal neuron? Then its transport. That how is it transported from the intestinal cell to its storage organ? And then we'll ultimately talk about the storage, that where it is stored. Okay. So it says retinal esters from animal sources and beta carotene from plant sources are absorbed from the intestinal lumen into the intestinal cell. So from the animal sources, you're getting retinol esters, and we know that from the plant sources, we get beta carotene, okay? So they are absorbed from the lumen into the intestinal cell. Now what is happening? They are then transported via chylomicrons and reach the liver, which is the storage organ. So your fat soluble vitamins, they are stored, and here the storage organ is liver. So they are transported as chylomicrons and ultimately reach liver. So all fat soluble vitamins are carried to storage organs via chylomicrons, which is very obvious. Why? Because they're fat soluble vitamins, so they need to be transported. They cannot be directly transported. They need to be transported via chylomicrons. Okay, because the chylomicrons, we all know that they are responsible for carrying exogenous tag. Therefore, fat soluble vitamins are also carried in chylomicrons. Okay, and the storage organ here, which is liver, it has receptors for ApoE, and this ApoE ligand, it is present in chylomicron. So your storage organ, it has ApoE receptors, and the ligand ApoE is present in chylomicrons. Then what is happening? The retinal esters are stored inside the liver in the perisinusoidal cells, that is known as ITO cells. When the body requires vitamin A, it is transported in the blood to the target organ with the help of two transport proteins forming a ternary complex, that is trimolecular complex. Now it is showing that in the storage organ, which here in this case is liver, in its perisinusoidal cells, your vitamin A is stored. Now, when the body requires vitamin A, then via the blood from the storage organ, that is liver, from the liver through the blood, it is transferred to the target organs. Your target organs are like your eyes, skin, where the action of vitamin A is taking place. Okay, the organs where action of vitamin A has to take place. Okay. Now, how is it transported? It is transported with the help of two transport proteins. Okay. So this is your vitamin A, and with the help of two transport proteins, these, so it forms a ternary, that is tri-molecular complex, as you can see, a ternary complex here. This you see, this ternary complex is formed. Now, the two proteins are transport proteins that we're talking about. They are retinol binding protein and transthyrectin. So, transthyrectin, if I break this name, trans is for transporter. Thy, because it is also the transporter for thyroid hormone. And rectin, because it is transporting retinol also. Okay, so that was about the absorption, transport, and storage. Now I'll quickly repeat this with the help of a handmade diagram in case any of you missed out something. Okay, so you can consider this to be the intestinal lumen, and this is your intestinal cell. And this is the intestinal cell. Now, what is happening through diet? Okay, now through diet, you are getting what you are getting from the plant source. You're getting carotenoids, mainly beta carotene. And from animal source, you are getting what? You're getting retinol esters. Now, what is happening? Your beta carotene, it is absorbed into the intestinal cell. Okay, now in the intestinal cell, it is acted upon by an enzyme known as dioxygenase, which will cleave the beta carotene into retinol. I had already shown you how beta carotene had two beta ion rings and retinol had single beta. Now this retinol ester that you're getting from animal sources, this retinol ester, it has what? It has your retinol, which is alcohol, and it has palmitic acid, which is fatty acid. Okay, retinol esters, how are they formed? They have your retinol plus fatty acid. Okay, now the fatty acid is removed. They are hydrolyzed. Okay, fatty acid removed, and what do you get? You get retinol. Now this retinol is absorbed into the intestinal cell. So in the intestinal cell, what do you have now? You have retinol and you have retinol, both from plant and animal sources. Now what is happening to this retinol? Fatty acid is added and it forms retinol ester. Now this retinol also, fatty acid is added and forms retinol ester. So ultimately you have retinol ester in the intestinal cell, and this retinol ester, then this is transported or carried to the storage organ, which is liver, via chylomicrons. Okay, very simple. And how are they transported? Uh, Via chylomicrons, like I said. Where are they transported? To the storage organ, which is liver, and from the liver, when need arises, body needs, they are transported through with the help of two transport proteins, that is transthyrectin and retinol binding protein. So that was about metabolism of vitamin A. Now moving on to the functions of vitamin A, very important. The most important function is vision. The form of vitamin A needed for vision is your 11 cis retinol. In the skin and mucosa, it is required for maintenance of epithelium. It is also required for reproduction, the retinol form. Then it is also involved in regulation of gene expression, which is the retinoic acid form. Then for growth and development, you need vitamin A for tissue differentiation. It also is an antioxidant, the most important being beta carotene. And then you have beta carotene. It also has photoprotective properties. Okay, therefore your beta carotene is applied in cutaneous photosensitivity. Okay, now moving on to vision. So 11-cis retinaldehyde, it combines with a protein known as opsin to form rhodopsin, which is also known as visual purple. So we'll be talking about Wald cycle. So for dim light, your rods are needed. Okay, so for vision in dim lights, you need rods, and these rods, they contain a pigment which is known as rhodopsin. And for colored vision, we all know cones are needed. Okay, so now we'll talk about the Wald's visual cycle. That's how your 11-cis retinaldehyde is combining with opsin and how you are able to see things. Okay, so here this is the cycle. Is it visible now? Okay. So what is happening? Let's see what is happening now inside the retina. Okay. So your 11-cis retinol, it changes into 11-cis retinol. Your all trans retinol, sorry, changes into 11-cis retinol. And then this 11-cis retinol changes into 11-cis retinaldehyde. Now this 11-cis retinaldehyde, it is going to combine with opsin, okay? The lysine residue in opsin, it is going to combine with that and form what? It's going to form rhodopsin, which is also known as visual purple. Now, this rhodopsin, also known as visual purple, it's when light falls on this, okay? You see, light is falling on this. So, when light falls on this, a series of photoisomerization reactions take place, okay? So, when light falls on visual purple or rhodopsin, a series of photo isomerization reactions take place. Okay, so your rhodopsin is converted into different isomeric forms like photorhodopsin, bathorodopsin, lumirhodopsin, metarhodopsin 1, metarhodopsin 2, and metarhodopsin 3. Now, this metarhodopsin 2, as you can see here, this is very important. Why is it important? Because this isomeric form, it is able to ignite signal cascade pathway. This metarhodopsin 2 is going to ignite okay, and then ultimately what is happening? So metarhodopsin 3 here, it is going to release opsin. You can see opsin is released and you have your all trans retinaldehyde here. Now this all trans retinaldehyde, it is again going to convert to all trans retinol. It converts to all trans retinol, and so you see it is going back to all trans retinol again here, this one. You see this arrow? This. So this is happening in the form of a cycle which is known as Wald's visual cycle. This is Wald's visual cycle. Okay. So I'll repeat this for you once again. What is happening in the retina? Your oil trans retinol, it is going to change into 11 cis retinol, then it changes into 11 cis retinaldehyde. In short, if you remember about the World's visual cycle, your 11 cis retinaldehyde is what is the form which combines with opsin to form rhodopsin. Now, this rhodopsin, when light falls on it, these photoisomerization reactions are only going to last a few fraction of a seconds. Okay, so a series of photoisomerization takes place, and you have different isomeric forms being formed of rhodopsin. That is your photorhodopsin, bathrhodopsin, lumirhodopsin, metarhodopsin 1, 2, 3. Now, this metarhodopsin to this isomer is important because it is able to ignite a signal cascade pathway. And the significance of igniting the signal cascade pathway, I'll just discuss later in the lecture. Then ultimately what happens? From your metarhodopsin 3, your opsin is released and you get all trans retinaldehyde. This all trans retinaldehyde again changes into all trans retinol, and this continues. So this is happening in a cyclic manner, and which is known as World's visual cycle. So this is how your vitamin A is involved in vision. Let's see what is metarhodopsin doing. Now this metarhodopsin 2, as you can see, it is able to activate this transducin. Okay, so your transducin, it is inactive and it is bound to GDP. Now this metarhodopsin 2, it is going to activate it, and this transducin is activated and it is now bound to GTP in active state. Now this transducin, which is GTP bound, it is able to activate your inactive phosphodiesterases. So your phosphodiesterases, they become active. Now what happens? These active phosphodiesterases, they are able to change cyclic GMP into 5 prime GMP. Now your 5 prime GMP, it is going to act on sodium channels and close them. Okay. This is exactly what the text says. We are going to read it. So the action of metarhodopsin 2, it is able to ignite a signal cascade pathway. Talking about that signal cascade pathway, let's see what is happening. The metarhodopsin 2 activates a G protein coupled receptor known as transducin. Okay, so your transducin is a G protein coupled receptor which is activated by metarhodopsin. Now transducin activates the enzyme. So the active transducin is going to activate phosphodiesterases. Okay, so your transducin, when it is in inactive form, it is GDP bound, and when it is active, it is GTP bound. Okay, so your active transducin, it is going to activate phosphodiesterases, and active phosphodiesterases, they convert cyclic GMP into 5 prime GMP. And 5 prime GMP acts on sodium channel and closes it. Okay, so now your sodium channels are closed. What will happen? This causes hyperpolarization. This causes hyperpolarization. Okay, now this hyperpolarization, it is going to generate nerve impulse. Usually what is happening, usually the action of action potential, it is brought about by depolarization. Okay, so usually action potential happens because of depolarization, but this is an exception where it is happening because of hyperpolarization. Okay, and a nerve impulse is generated. So that was about your World's visual cycle and the signal cascade pathway, which was activated by metarhodopsin 2. Now, talking about regulation of gene expression. So your vitamin A, it helps in growth and development and also in tissue differentiation. Retinoic acid is an important form. Retinoic acid, it binds to nuclear receptors and activates it. Okay, the active nuclear receptor then binds to response element of gene and regulate gene expression. Retinoic acid acts via nuclear receptor just like steroid hormones. So in signal transduction, you must have studied the action of steroid hormones, how they are binding to nuclear receptors and not surface receptors that are present on the membrane. So your vitamin A, they also, your retinoic acid work in the similar fashion. I'll show you how. your retinoic acid, which is going to bind to nuclear receptors. Okay. Now, active nuclear receptors, they are going to bind to response elements on the gene. Okay. Now, this will regulate gene expression. Okay. Now, there are two types of receptors, it says RAR and RXR. Let's study the difference between two receptors, these two receptors. So, your RAR, or also known as retinoic acid receptors, the form of retinoic acid that acts as ligands for RAR is all trans retinoic acid. So your all trans retinoic acid, it is going to act via RAR receptors. Active RAR receptor, it binds to response elements and regulate gene expression. So this is very simple. Now talking about RXR, that is retinoic acid X receptor. So let's see how is it different from the RAR receptor. The form of retinoic acid that acts as ligand is 9-cis retinoic acid. Okay, so here your 9-cis retinoic acid is going to act via RXR, and your all trans retinoic acid, they act via RAR. Now RXR, it dimerizes with active vitamin D receptor. Okay, it can also bind to thyroid hormone receptor. Now this heterodimer bind to gene and regulate gene expression. Thus vitamin D deficiency can also cause vitamin A deficiency, and similarly thyroid deficiency can also lead to impairment of vitamin A function. So the difference between the two receptors, as we gather from the information given, is that, let's suppose this is your RAR receptor, and for RAR it says that the ligand is your all trans retinoic acid. So your all trans retinoic acid, it binds to RAR, and your RAR nuclear receptor, it is activated, then it is going to bind to the response element next to the G, and regulate gene expression. Now talking about the RXR receptor, let's suppose this is the RXR receptor. So the ligand for this is your 9-cis retinoic acid. This is 9-cis retinoic acid. Okay. So 9-cis retinoic acid binds to RXR. Now this RXR cannot directly bind to the response element. First, it dimerizes. It forms a heterodimer with an active vitamin D or thyroid hormone receptor. So this is your RXR having the ligand all cis retinoic acid, and it is going to dimerize with a active vitamin D receptor. Okay, vitamin D receptor, and this is your vitamin D. So this is vitamin D, and this is vitamin D receptor. Now this heterodimer, it is going to bind to the response element and regulate gene expression. So this is the difference between RAR and RXR receptors. Now talking about the deficiency of vitamin A. So in the eye, there's loss of vision to green light, nyctalopia, which is night blindness, then dryness in the conjunctiva and cornea. This is known as dry eyes or xerophthalmia. Keratitis, which is inflammation of the cornea. Then you have Bittwort spots, corneal ulcers, which is also known as keratomalacia. Your vitamin A deficiency is the most common vitamin deficiency, and it is also the most common cause of preventable blindness. I'm going to be a little fast now because we're running out of time. Okay. So in the skin, the manifestations are seen as follicular hyperkeratosis. So for the maintenance of epithelium, you need vitamin A. It is also known as papillary dermatitis or toad skin. Now, toxicity of vitamin A, it is usually toxicity is seen in Arctic explorers because your Arctic explorers they depend on polar bear for food. Okay, polar bear. So they consume the liver of polar bear, which is the storage organ of vitamin A, which leads to the toxicity of vitamin A. And the organelle damaged in hypervitaminosis is your lysosome. Okay, so the toxicity is manifested as headache, nausea, and hip fractures. Hip fractures, why? Because your vitamin A is increased, and when vitamin A is increased, it increases osteoclast activity. So, increased osteoclast activity leads to increased resorption, and which leads to increased fracture susceptibility. Okay. A therapeutic applications, talking about beta carotene, it is photoprotective, therefore used for cutaneous photosensitivity. Talking about all trans retinoic acid, it is used for mild acne or tretinoin. It is used to prevent skin sagging. It is not teratogenic. Okay. And your 11th is retinoic acid, which is isotretinoin. It is used for cystic acne. It's also used in childhood neuroblastoma. And it is teratogenic. Therefore, we say that it is not pregnancy and lactating safe. The sources in RD of vitamin A, your retinoids, you get them from animal sources like liver, egg, butter, cheese, milk, fish, and meat. And carotenoids, they are derived from plant sources. The richest source of beta carotene is your carrot, mangoes, papaya. And then RDA in children, the range is 400 microgram per day. In pregnancy, you need 800 microgram per day of vitamin A. In lactation, 950, and in men and women, 600 microgram per day. So that was about vitamin A. Quickly moving on to vitamin D. So your vitamin D, it is a group of sterols which has hormone-like action. It is also known as sunshine vitamin, very important. So, which is your sunshine vitamin? It is your vitamin D. The compounds considered as vitamin D, you have ergocalciferol, which is from plant source, that is vitamin D2, and D3, which is cholecalciferol. You get them from animal sources. So, these are the active forms of vitamin D. Now, sources of vitamin D, endogenous vitamin precursors. So, basically, they are endogenously synthesized. How? When light, when UVB rays, okay, UVB rays in 290 nanometer, in the range 290 nanometer to 315 nanometer, they fall on skin. Okay, you see UV rays when they fall on the skin in the range of 290 nanometer to 315 nanometer, photoisomerization reactions take place inside skin. So photoisomerization reactions take place inside skin, and ultimately what is formed? Your D3, that is cholecalciferol is formed. So light falls on the skin, your 7-dehydrocholesterol, it is converted into through a series of photoisomerization reactions into cholecalciferol. Now, through diet, all diet is poor in vitamin D unless fortified. That is, unless artificially added, all diet is poor in vitamin D. Vitamin D, usually it is synthesized in the body. A preformed vitamin D is a requirement only in individuals with limited exposure to sunlight. Okay, so those people who are not exposed to sunlight, then they need to take vitamin D from outside. Otherwise, it is endogenously synthesized in the body. Okay, so the only source of vitamin D is fish and food. Unless it is fortified, it is a poor source of vitamin D. Now, what is happening from your diet, which is a minor source, major source being synthesis in the skin. Okay, so from your diet, you get ergocalciferol, which is from plants like ergots and from animal sources, you get cholecalciferol. Okay, so cholecalciferol is from animal sources like fish, and ergocholecalciferol, it is from plant sources. So your ergocalciferol, it is also finally changed into cholecalciferol. Okay, and from animal sources, you're directly getting cholecalciferol, and then in the skin also 7-dehydrocholesterol, it is changing into cholecalciferol. So ultimately, you have cholecalciferol in your body. Okay, and this is also known as calciol. Cholecalciferol also known as calciol because it has one hydroxyl group. It is having one hydroxyl group. Okay. Now moving on to the metabolism of vitamin D. So through diet, which is very less amount, majorly it is synthesized in the body, or through endogenous synthesis, we have cholecalciferol, that is calciol, inside our body. I just explained you how. Now, with the help of transport proteins, that is vitamin D binding proteins, cholecalciferol reaches liver. So, through diet or whether it being endogenously synthesized, ultimately you are ending up with cholecalciferol, that is calcium, in your body. Now, what is happening? With the help of transporters, this cholecalciferol, it reaches liver. Now, in the liver, what is happening? Via the action of the enzyme 25 hydroxylase, which hydroxylates, that is, adds a hydroxyl group to this cholecalciferol at the 25th position and becomes calcidiol, because now it is having two hydroxyl groups, therefore it becomes diol. That is one that was initially present and the another one at 25th position. So 25 hydroxy calci cholecalciferol or 25 hydroxycalciol or calcidiol. Now this was happening in the liver. Now what is happening? This calcidiol from the liver, it is transported in the kidney, and in the kidney another hydroxylation reaction is taking place via the action of the enzyme alpha 1 hydroxylase. Okay, now, this alpha 1 hydroxylase, it is also favored by parathyroid hormone and is considered as the rate limiting enzyme. Now, what is happening? This calcidiol in the kidney, it is converted into calcitriol. So, this calcitriol is the biologically active form of vitamin D. Now, let's understand this with the help of a diagram. It will become easier for you. So, either in the skin, as you can see, you have cholecalciferol being synthesized, or either coming from the diet. Now, this cholecalciferol is transported to the liver. In the liver, what is happening? The action 25 hydroxylase is taking place and calcidiol is formed. Now, this calcidiol, it reaches the kidney. In the kidney, what is happening? Calcitriol, which is the active form of vitamin D, is being formed. The enzyme is alpha 1 hydroxylase. Now, this alpha 1 hydroxylase, it is favored by parathyroid hormone. And this parathyroid hormone, it is favored by low calcium. Okay. Now it says when the body does not require vitamin D, it is converted into inactive form by 24 hydroxylation. So your 24 hydroxylation, it is going to inactivate vitamin D. So in the kidney, if 24 hydroxylation is taking place, what is formed? Calcitetrol is formed because 3 hydroxyls were already present, another being added at the 24th position, and you end up with calcitetrol. Now, in the liver, if the 24 hydroxylation is taking place, you end up with 24 hydroxycalcidiol. Calciol is cholecalciferol, calcidiol is 25 hydroxycholecalciferol, calcitriol is 125 dihydroxycholecalciferol, and calcitetrol is 24 hydroxylation of calcitriol. Okay, so if you've understood this, you will not get confused between these names. We're talking about the functions of vitamin D. So your vitamin D, it is involved in regulation of calcium and phosphorus. It has immunomodulatory function. It also has anti-proliferative role. During bone development also vitamin D is required for bone mineralization. We're talking about the regulation of calcium and phosphorus. So the three main factors that are responsible for the maintenance of calcium level in the body, it is vitamin D, the parathyroid hormone, and calcitonin. And at the sites where the regulation is brought about, it is the intestine, kidney, and the bones. So let's see the effects of these three factors. So your calcitriol, it increases serum calcium and it increases serum phosphorus. Parathyroid hormone, it increases serum calcium, but on the other hand, it decreases serum phosphorus. And your calcitonin, it decreases serum calcium. So calcitonin, for the sake of remembering, you can remember it in this way. A tonin, okay? So it is a tonic. It is a tonic for the bone. Calcitonin, you can remember it in this way. It is a tonic for the bones. For the bone, so it will deposit calcium in the bone. If it is going to deposit calcium in the bone, definitely it is going to decrease the serum calcium levels. Okay. Now let us see how these actions are being brought about. So the action of vitamin D and parathyroid hormone in the intestine. So your parathyroid hormone, as we already studied, it favors alpha-1 hydroxylase, which increases the level of active vitamin D. Alpha-1 hydroxylase, it will hydroxylate to calcidiol and form calcitriol. So your calcitriol, which is the active form of vitamin D, this will be increased. Now this calcitriol, it is going to increase the level of a particular calcium transport channel and a calcium binding protein which increases the absorption of calcium from intestine. So your calcitriol, it is going to increase the expression or the level of a particular calcium transport channel and a calcium binding protein which are responsible for increased absorption of calcium from intestine. When the absorption of calcium from the intestine is increased, obviously the level of serum calcium will also be increased. Now, action of vitamin D in the kidney. In the kidney, what is happening? Your vitamin D, that's it, calcitriol, it increases reabsorption of calcium and phosphorus. In the kidney also, it is increasing the reabsorption. In the intestine, it is increasing the absorption. In the kidneys also, it is increasing the reabsorption. Again, this is how the serum level is increasing of calcium and phosphorus. We're talking about the action of parathyroid hormone in kidneys. Parathyroid hormones favors reabsorption and causes phosphorus excretion. Okay. So calcium reabsorption is favored, but excretion of phosphorus is taking place. Thus, as a result of parathyroid hormone, it increases serum calcium but decreases serum phosphorus because phosphorus is being excreted. We're talking about the action of vitamin D and parathyroid hormone in bones. So both parathyroid hormone and vitamin D, they have the same action. So we'll take them together. What is happening is that your osteoblasts, they have a rank ligand, okay? And the receptor for rank ligand is present in the pro-osteoblast. So suppose this is your osteoblast, and your osteoblast, it is having a rank ligand. Okay, and the receptor for this rank ligand is present in the pro-osteoclast. So the receptor here, this is present in pro-osteoclast. Okay, now what is happening? The parathyroid hormone and vitamin D, it increases the action of this rank ligand. Okay, this is the rank ligand. This is the receptor for rank ligand. This is your osteoclast, or osteoblast, sorry, and this is your osteoclast. Now what will happen? They will bind to it, and what happens? This action is increased by vitamin D and parathyroid hormone. Now this favors the conversion of this pro-osteoclast. This pro-osteoclast, it is converted into mature osteoclast. Okay, mature osteoclast. Now this mature osteoclast, it is going to bind to bones and lead to bone resorption. It is going to bind to bones and lead to bone resorption. Okay, thus vitamin D and parathyroid hormone increases serum calcium by increasing osteoclast activity, which causes bone resorption. So action of calcitonin in bones is just the reversal. It decreases serum calcium by decreasing osteoblast activity. So calcitonin is going to increase the osteoblast activity. So all the calcium is seen inside the bone. Whereas your vitamin D and parathyroid hormones, they were converting, increasing the rank ligand, which was converting the pro-osteoclast into mature osteoclast. So the osteoclast activity was increasing and bone resorption was increasing. Therefore, serum calcium was increasing because there was increase in osteoclast activity. Moving on to the immunomodulatory action of vitamin D. The antibacterial action of vitamin D can be seen in prevention of Mycobacterium tuberculosis. So apart from kidneys, conversion of calcidiol to calcitriol is also taking place in the macrophages. So we've seen that your calcidiol was converted into calcitriol in the kidneys. But now it says that apart from kidneys, it is also taking place in macrophages by cytochrome 27B. So I'll explain you with the help of a diagram. Imagine this is a macrophage. Okay, this is a macrophage. This macrophage, it has toll-like receptors. Okay, to these toll-like receptors, your Mycobacterium tuberculosis comes and binds. Okay, now it is saying that apart from the kidney, your calcidiol was being converted into calcitriol even in the macrophages. Okay, so 25 hydroxy calcidiol, it was being converted into 125. Okay, so your calcidiol being converted into 125 dihydroxycalcidiol or calcitriol. Okay, now what is happening? And this was brought about by, it says, cytochrome 27B. Okay, now when Mycobacterium tuberculosis comes and binds to toll-like receptors, it is going to stimulate or activate cytochrome 27B, and cytochrome 27B will convert more calcidiol into calcitriol, so your calcitriol level will increase. So this calcitriol level is increased, and this calcitriol, it is going to activate cathelicidins. These are antimicrobial peptides and they are going to kill the bacteria. So that was talking about the immunomodulatory action of vitamin D. So it says, so the level of calcitriol increases inside a macrophage. When Mycobacterium tuberculosis binds to toll-like receptors in macrophages, it increases the activity of cytochrome 27B, which in turn converts calcidiol to calcitriol. An increased level of calcitriol inside macrophages, they favor the production of cathelicidin, which is an antimicrobial peptide from defensin family. And your cathelicidin is going to kill the bacteria. And thus, this explains the neuromodulatory action of vitamin D. Now, talking about vitamin D deficiency, very, very important. So, it causes demineralization of bones, leading to rickets in children and osteomalacia in adults. So, your rickets is very important, very frequently asked. Okay. Now, talking about the clinical features of rickets. So you have bone deformities in this. Your bones become soft and pliable. Weight-bearing bones are bent. There is bow legs, knock knees, ricti rosary. Ricti rosary is nodules formed in the costochondrial junction. Okay, there's bossing of frontal bone, pigeon chest, Harrison sulcus, also known as Harrison groove. Now you can see these diagrams. Bow legs, you can see the legs are in the form of a bow. Knock knees, the knees are together and the legs are apart. These are knock knees. Then you can see ricti rosary, nodules in the costochondrial junction. Then you can see this Harrison sulcus. This is due to softening of ribs. Now talking about the types of rickets, there are various types. Here I've taken the two most important types, that is nutritional rickets and renal rickets. So in nutritional rickets, there's insufficient exposure to sunlight or deficiencies in vitamin D consumption occur predominantly in infants or elderly. Vitamin D deficiency is more common in northern latitudes where there is not much sunlight. Renal rickets in kidney diseases, even if vitamin D is available, calcitriol is not synthesized. Okay, since your alpha 1,4 hydroxylase, 1 alpha hydroxylase, that is the calcidiol being converted into calcitriol, it was taking place in the kidney. So even if everything is fine, but there is some defect in the kidney, in renal rickets, there is some defect in the kidney. So because of your kidney disease, your active calcitriol, that is vitamin D, is not being synthesized. So therefore, this responds to administration of calcitriol. We're talking about osteomalacia. It is seen in adults. The bones become soft due to decreased mineralization and it is more prone to fractures. Talking about toxicity of vitamin D. Excess vitamin D gets deposited in soft tissues and blood vessels known as calcilosis. It causes contraction of blood vessels leading to hypertension. Toxicity is seen only with supplements. Now, excess vitamin D produced in the skin is converted to inactive form. Now, talking about the RDA in children, you need 10 mg per day. In adults, 10 to 5 mg per day. And in pregnancy, 10 mg per day of vitamin D is required. So with this, we come to the end of vitamin D. I think we can take vitamin E because there's not much in vitamin E. Vitamin K, I think I'll take vitamin K before we begin with the next class. Okay. So vitamin E, not really very important and not many questions based on this. I hope the screen is visible. Okay. So vitamin E, we'll quickly take this. Just talking about vitamin E is stereoisomer of tocopherols. Tocopherol has different stereoisomers out of which the alpha tocopherol is the most potent and active form. So vitamin E has chromane ring as you can see here and it has an isoprenoid unit. Okay, now the alpha tocopherol which is an isomer, the tocopherols they have various isomers, different stereoisomers in which the alpha form is the most potent vitamin E. Now, talking about the functions of vitamin E, it is naturally occurring and most potent chain breaking antioxidant vitamin. It prevents oxidation of PUFA, LDM, and therefore it reduces the oxidative stress in the body. So, the most important function of vitamin A is that it is the chain breaking antioxidant in the body. Thus, therefore, it is involved in reducing the oxidative stress in the body. Talking about the deficiency of vitamin E, vitamin E deficiency is seen in patients who cannot absorb dietary fat, in premature infants, and in A beta lipoproteinemia and in mutations in the gene for tocopherol protein transfer. Now, clinical manifestations, vitamin E deficiency causes neurological problems due to poor nerve conduction. These include neuromuscular problems like spinocerebellar ataxia, retinopathy, peripheral neuropathy, skeletal myopathy. vitamin E deficiency also causes hemolytic anemia. Why? Because we all know that I've just told you that it is an antioxidant, okay, a chain breaking antioxidant. So it is involved in free radical scavenging, and free radicals are able to damage the cell membrane, okay. So to maintain the integrity of RBC membrane, you should not have free radicals. So in case of vitamin E deficiency, there is hemolytic anemia. But talking about toxicity of vitamin A, it is rare, but if present, it is the least toxic vitamin, fat-soluble vitamin. Sources of vitamin E, they include vegetables, oil, they are rich in vitamin E, such as sunflower and safflower. Liver and eggs are a moderate source of vitamin E. Talking about the RDA in adults, 15 mg per day is required, and requirement of vitamin E increases as the intake of polyunsaturated fatty acid increases to limit fatty acid peroxidation. So that was briefly about vitamin E, not very important and almost never really asked. Okay. So with this, we come to the end of the lecture. Vitamin K I'll take before I begin with the B complex. Vitamin K is also not very long. I think it'll take around 5 to 7 minutes. Okay. So if you have any queries, you can send them before I end the class. Okay. Thank you for watching. See you in the next class.